Imagine your brain not just as an organ or a biological computer, but as this massive, sprawling, bustling metropolis. And in this incredibly dense city, you have roughly 86 billion residents.
86 billion. It's a staggering number when you really sit with it.
Right. And these residents, they're your neurons. They are constantly talking to each other, coordinating literally every thought, every heartbeat, every fleeting memory, every movement. And they do this using a highly specific lightning-fast electrical language. It is a city of perfect order, governed by strict traffic lights and precise communication lines.
Very delicate balance.
Exactly. But what happens when a single chemical compound derived from a plant, specifically delta-9 tetrahydrocannabinol, or THC, from cannabis sativa, floods into this city? Suddenly the traffic lights change. The communication lines are completely rewired. And even your very perception of the flow of time itself begins to stretch and warp.
It truly is a profound biological takeover. And the reason it works so seamlessly is because this plant compound is essentially picking a highly sophisticated lock that already exists within the human brain. The lock was never designed for the plant. The plant just happened to evolve a molecular key that fits it perfectly.
I love that visual. A master key that just happened to grow on a plant. And that is exactly the mission of our deep dive today. We are going straight into the source material to explore the profound, intricate neurobiological journey of THC in the human brain.
Yeah, and we're not just going to talk about what happens during a high in some vague, abstract sense. We are going to really explore the microscopic architecture of neural communication.
We'll look at how cannabis literally hacks an ancient biological circuit breaker. We will examine the neuroadaptations that occur with chronic use, and I think most importantly, we're going to trace the incredible timeline of homeostatic recovery when the brain is left to heal and recalibrate.
It's a journey that takes us from the absolute baseline of how a single neuron fires all the way to the complex cognitive restoration of executive functions. And to do this, we'll be looking closely at some fascinating studies. We've got everything from high-resolution KT imaging of the brain's receptors to detailed behavioral assessments, like classical eye blink conditioning.
We have a ton of ground to cover. So whether you are intensely curious about neurobiology, perhaps you're studying addiction, or maybe you just want to know exactly why time seems to slow down to a crawl when someone consumes cannabis, this deep dive is going to give you the complete picture. We're taking you from the very first action potential to the final stages of cognitive restoration.
To truly appreciate the magnitude of the THC hack, we first have to establish what normal looks like. We have to look at the baseline brain and really understand the foundations of the action potential.
Right, because every single function of the brain relies on neurons communicating across a tiny gap called the synapse.
Exactly. But before a chemical message can even be sent across that gap, an electrical spark has to travel down the neuron itself.
I was actually amazed looking at the research on how neurons rest. I always pictured them just sort of sleeping or sitting dormant when not in use. But they aren't dormant at all, are they? The neuronal membrane is described as this highly regulated, incredibly selective barrier.
Yeah, the term resting state is almost a misnomer because it implies a lack of activity. In reality, inside the cell, there is a negative charge of exactly negative 70 millivolts compared to the outside.
Negative 70 millivolts.
Right. And this negative charge is not some biological accident. It is a state of intense, carefully maintained readiness.
It's like a drawn bowstring, just waiting for the archer to let go.
That is the perfect analogy. And keeping that bowstring drawn requires a massive amount of energy. That state of readiness is maintained by one of the absolute hardest working mechanisms in the body. It's called the sodium-potassium pump.
The housekeeper.
The housekeeper, yes. Or maybe the power grid manager of our cellular metropolis. It constantly uses ATP, which is the primary energy currency of the cell, to actively pump 3 sodium ions out of the neuron for every 2 potassium ions it brings in.
So it's constantly kicking out more positive charge than it's letting in, which is exactly how the inside stays negative at that negative 70 millivolts. But the amount of energy that takes, I read that something like 20 to 40% of the brain's entire energy budget goes just to this one pump, just to maintain the resting state.
It is staggering when you think about it. The brain consumes about 20% of the body's total energy, and a huge fraction of that is just spent keeping these billions of bowstrings pulled back, working against the natural flow of diffusion.
Okay, let's unpack this, because the moment of action is where it gets explosive. When this neuron receives excitatory signals from its neighbors, the voltage starts to rise, right? It creeps up from that negative 70 millivolts until it hits a very specific threshold.
Yes, usually around negative 55 millivolts. And the second it hits that specific threshold, it's like a dam breaking. Voltage-gated sodium channels along the membrane just fly open.
And all that sodium that the housekeeper pump has been painstakingly pushing out suddenly rushes back in.
Because it wants to balance out the charge. Both the electrical gradient and the concentration gradient are pulling it inside, and this is depolarization.
Depolarization.
Got it. Precisely. The rush of positive sodium ions spikes the internal voltage of the cell all the way up to positive 30 or even positive 40 millivolts in just a fraction of a millisecond. That is the actual spark. That's the action potential.
But it can't stay there.
No, the neuron cannot stay in that highly positive state. Almost immediately, those sodium channels slam shut and a different set of doors opens, the potassium channels. Potassium, which is heavily concentrated inside the cell, rushes out, carrying its positive charge away to restore the negative environment.
Repolarization.
Exactly. And it even overshoots for a moment, dropping below negative 70 in a phase called hyperpolarization before the housekeeper pump kicks back into high gear to sweep the sodium back out and reset the whole system.
And this entire cycle, this spark, is an all-or-none event, correct? Like flipping a light switch. There is no such thing as a half fire.
That's right. It fires or it doesn't. And the reason this microscopic electrical storm matters is because this action potential travels down the length of the neuron's long cable-like axon until it reaches the terminal.
And that's where the chemistry takes over.
Yes. Once there, the electrical signal triggers the influx of calcium. Calcium acts as the final trigger, forcing these tiny bubbles, called vesicles, which are full of chemical neurotransmitters, to fuse with the membrane and spill their contents into the synapse, that gap between neurons.
So the electrical spark inside the city's wires turns into a chemical ferry crossing the river to the next building.
Beautifully put. This chemical message floats across the gap and binds to receptors on the next neuron, telling it to either fire its own action potential or stay quiet. This is the forward-moving, highly polarized language of the brain. It is literally how you think, how you move, and how you feel.
But the brain is incredibly dense. The city is packed. If neurons were just constantly firing these action potentials, shouting at each other with excitatory neurotransmitters like glutamate, wouldn't the whole system just overheat? I mean, it seems like it would be wildly overstimulated. There has to be a way to regulate this constant firing so the city doesn't literally short circuit.
And that profound need for regulation brings us to the discovery of the endocannabinoid system, or the ECS. When researchers first started piecing the system together, they realized they were looking at a master regulator of homeostasis. Its entire evolutionary purpose is to keep the physiological systems of the brain and body within a very narrow, safe, healthy operating range.
This is one of my absolute favorite concepts from the source material. Because normally, as we just established, the flow of traffic is strictly one way. Neuron A sends a message forward to neuron B. But the ECS operates entirely in reverse. It utilizes something called retrograde signaling.
What's fascinating here is that for the longest time, the scientific consensus, the absolute dogma in neuroscience, was that synaptic transmission was strictly a one-way street, forward only.
Right, nobody even considered backward signaling.
Exactly. But researchers in the early 1990s studying regions like the hippocampus and the cerebellum were completely stunned. They discovered that neuron B, the receiving neuron, could actually talk back to neuron A to shut it down.
It was like watching cars successfully drive backward on a one-way highway. So how exactly does neuron B do that? Let's say neuron B is getting absolutely hammered by excitatory signals. Neuron A is just shouting glutamate at it nonstop. To protect itself from overheating, neuron B creates endocannabinoids directly on demand, right from the fat, the lipids in its own cell membrane.
Yes, and that on-demand aspect is crucial. They aren't stored in little vesicles waiting to be released like normal neurotransmitters. Because they are lipids, fat-soluble, they would just slip right through the vesicle walls anyway. They are synthesized exactly when and where they are needed.
And what are these molecules called?
The two main endocannabinoids your brain produces are 2-arachidonylglycerol, which we usually just call 2-AG, and anandamide.
I love the history behind that naming. Anandamide gets its name from the Sanskrit word ananda, which translates to bliss or divine joy. It's the bliss molecule.
It is a very fitting name for a molecule that induces relaxation and balance. Once neuron B synthesizes these endocannabinoids, because they are fat-soluble, they can simply slip right through the watery environment of the synaptic cleft. They diffuse backward, and they travel in reverse to bind to specific receptors located on the presynaptic terminal of neuron A.
These are the famous CB1 receptors.
The very same.
And when anandamide, or 2-AG, binds to that CD1 receptor on the shouting neuron A, it acts exactly like a biological circuit breaker. It cuts the power.
It is a remarkably elegant and highly efficient circuit breaker. To look closely at the molecular mechanics for a moment, CB1 receptors belong to a family called G-protein coupled receptors. Specifically, they are coupled to what we call the GI/O subclass.
Okay, let's translate that for a second so we don't get lost in the alphabet soup. When you say it's a G-protein coupled receptor of the GIO subclass, what does that actually mean for the neuron?
The I stands for inhibitory. When the endocannabinoid binds to the CB1 receptor on the outside of the cell, it changes the shape of a protein on the inside of the cell. This triggers a cascade. It inhibits an enzyme called adenyl cyclase, which basically lowers the cell's energy messengers.
It's calming it down.
Exactly. But most importantly, it acts like a bouncer at a club. It blocks the voltage-gated calcium channels from opening, while simultaneously opening the potassium doors to let positive charge out.
And if we remember from a minute ago, calcium is the final trigger that forces the neuron to release its chemical messengers. So no calcium coming in means absolutely no neurotransmitters can be released. Neuron A is effectively silenced. The shouting stops, the circuit breaker flips, and the brain city is protected from overstimulation.
Exactly. And then, almost as quickly as they were created, these natural endocannabinoids are swept away. They are broken down by enzymes like FAAH, fatty acid amide hydrolase. They do their job, they restore the balance, and they vanish within fractions of a second. It is a localized, temporary, and highly precise system of self-regulation.
Which brings us to the core of this entire deep dive, the THC hack. If the brain's natural system is so elegant and localized, how does the primary psychoactive component of a plant fit into this delicate, precisely timed system?
THC is what pharmacologists call a partial agonist at these exact same CB1 receptors. Its molecular structure happens to mimic the brain's natural endocannabinoids just perfectly enough to turn the key in the lock.
But the delivery method is entirely different, right? The brain's natural molecules, 2-AG and anandamide, are released at a single specific synapse for a fraction of a second, exactly when a specific circuit is overheating. But when someone consumes cannabis, THC enters the bloodstream and floods the entire city simultaneously.
That is the critical distinction. It doesn't wait for a neuron to become overexcited. It doesn't target just the synapses that need regulating. It binds indiscriminately to CB1 receptors everywhere in the brain all at once.
And because it's a foreign molecule, it isn't broken down quickly by those cleanup enzymes like FAAH.
No, it isn't. It stays locked into the receptors for hours.
So it's like someone broke into the city's power grid and just started randomly pulling every circuit breaker they could find.
Precisely. By binding everywhere at once, THC causes a widespread, uncoordinated suppression of neurotransmitter release. You see, the brain relies on a very specific signal-to-noise ratio to function. It needs certain circuits to be actively firing and others to be completely quiet so that clear information can be processed. THC effectively flattens this ratio. It muddies the waters.
To really understand what that flattening means for you, the listener, we need to connect this microscopic mechanism to the actual subjective experience.
Yeah.
If we take a tour of the brain on THC, we can see exactly why the high feels the way it does. Instead of just listing off brain regions, let's look at how this widespread hacking alters your reality chronologically.
That's a great approach. Let's start with the cerebral cortex.
Right, the cortex is packed with CB1 receptors. This is the outer layer of the city, responsible for your complex, higher-order thinking and sensory perception. When THC hits here, sensory inputs get scrambled. Colors might seem brighter, music might sound more profound, but your executive function takes a massive hit.
Yes, the cortex is where you process the immediate world. But the scrambling of the cortex alone doesn't explain one of the most famous effects of cannabis: the sudden loss of train of thought. Have you ever been talking to someone who has consumed cannabis and right in the middle of a sentence they just stop and say, wait, what was I just talking about?
All the time. It's like the previous 10 seconds were just wiped from a hard drive.
That phenomenon takes us deeper into the brain, to the hippocampus, which is critical for memory formation. The sources detail how the hippocampus relies on highly synchronized electrical patterns called sharp wave ripples and theta oscillations.
Oscillations, like a rhythm.
Think of these as the rhythmic ticking of a clock that allows the brain to encode short-term memory and establish the sequential order of events.
And when THC binds to the CB1 receptors in the hippocampus, it profoundly disrupts these oscillations. It essentially shuts down the recording equipment. So it impairs the encoding of memory so severely that the user loses what are called their temporal anchors, the sequential markers of what just happened. You forget the beginning of the sentence because the hippocampus never successfully hit save on the concept.
Exactly. And as the user is trying to navigate this scrambled memory, they might also notice a change in their physical coordination. This takes us to the cerebellum and the basal ganglia, the brain's motor control and timing centers.
Right, flooding these areas with THC impairs balance and fine motor skills. The sources highlight some absolutely fascinating research on something called classical eye blink conditioning to prove this point.
Yes, let's talk about the eye blink conditioning. This is a brilliant experimental model. Classical eye blink conditioning is a very specific type of learning dependent entirely on the cerebellum. A subject is trained to blink in response to a tone that predicts a puff of air to the eye.
So the tone plays and they learn to blink right before the air hits.
Exactly. It requires precise microscopic timing. Studies show that chronic cannabis users exhibit markedly fewer and much more poorly timed conditioned responses compared to non-users.
They just can't coordinate the timing of the blink to match the tone.
Right. And what's amazing is that this maps perfectly onto findings in mice that have had their CB1 receptors genetically knocked out. It shows a literal impairment in the circuitry that calculates timing and motor response directly linked to the endocannabinoid system.
Okay, so your thoughts are a bit scrambled, your memory is dropping frames, your physical coordination is slightly delayed, and then invariably you find yourself scaring into the pantry. Let's talk about the munchies. Because suddenly a stale bag of tortilla chips seems like a culinary masterpiece. Why does THC do that?
That takes us to the hypothalamus, the brain's energy regulation center. Normally, when you've eaten enough, the hypothalamus releases a peptide called cholecystokinin, or CCK. CCK is the chemical signal for satiety. It tells your brain, we are full, stop eating. THC actually inhibits the release of CCK. It silences the fullness signal while simultaneously stimulating the pro-appetite pathways. You aren't just psychologically craving food, your brain is being chemically instructed that you are starving.
That is wild. The circuit breaker shuts off the I'm full alarm. But there is another, much more serious emotional effect we have to cover. The amygdala, the brain's emotional core.
This is a critical one.
THC's interaction here is incredibly complex. For some, it causes profound relaxation and euphoria. But for others, or particularly at high doses, it can trigger intense anxiety, fear, and paranoia. Why is it so unpredictable?
It comes back to the fact that CB1 receptors are located on both excitatory glutamate neurons and inhibitory GABA neurons. In the amygdala, THC can inhibit the release of GABA, which normally keeps fear responses in check.
So GABA is usually calming the amygdala down.
Exactly. If you turn off the inhibitory brakes in the fear center, you get runaway anxiety and paranoia. It's a complex disruption throwing emotional processing entirely out of balance, and it varies heavily based on the individual's baseline neurochemistry and the dose of THC.
It really is the widespread, indiscriminate nature of this receptor activation that makes the THC experience so multifaceted. However, if we want to understand why cannabis is inherently rewarding, why people seek it out despite the memory impairments and the potential paranoia, we have to look closely at the dopamine connection. Dopamine is the system that assigns value, motivation, and salience to our actions.
It is the currency of reward, and this is vital to understand.
Here's where it gets really interesting, because the way THC triggers dopamine isn't direct. It isn't like other substances that just force dopamine out. It's a game of neurobiological dominoes. Let's look at the mesolimbic reward pathway, which is the brain's primary reward circuit, connecting the ventral tegmental area, or VTA, down in the midbrain, to the nucleus accumbens in the forebrain.
To use your Metropolis analogy, the VTA is the power plant generating the reward signal, and the nucleus accumbens is the downtown center receiving it. In a normal baseline state, the dopamine-producing neurons in the VTA are kept under incredibly tight control.
They aren't just firing wildly.
Not at all. There are local interneurons right next to them that constantly release GABA, the brain's main inhibitory neurotransmitter.
So these GABA neurons are acting like a constant brake pedal on the dopamine neurons. They keep the dopamine power plant idling, firing at a very low, steady rate of about 1 to 5 hertz, just enough to keep you functioning, but not enough to make you feel euphoric.
Precisely. But those GABA interneurons are absolutely covered in CB1 receptors. So when THC floods the VTA, it binds to the CB1 receptors on those GABA neurons and activates the circuit breaker. The release of GABA is immediately stopped.
By inhibiting the inhibitor, THC effectively takes the foot off the brake pedal, or literally cuts the brake lines. And without that GABA brake, the dopamine neurons are completely disinhibited. They start firing rapidly in high-frequency bursts, flooding the nucleus accumbens with massive waves of dopamine.
This surge of dopamine in the nucleus accumbens creates a powerful sense of reward. It signals to the brain that whatever experience is happening right now has extraordinarily high motivational value. This drives the reinforcement loop that can lead to continued habitual use.
But the dopamine story doesn't end there, does it?
No, it doesn't. What is truly groundbreaking in recent research is the realization that the reward story isn't confined entirely to the midbrain. There is another critical mechanism at play involving the cortex and completely different type of brain cell altogether,
Yeah.
the astrocyte.
Yes, astrocytes. These are star-shaped glial cells. And for the longest time, scientists used to think they were just the structural support of the brain. The word glia literally comes from the Greek word for glue. We thought they just had the neurons in place.
That was the dogma for decades. We thought neurons did all the thinking and astrocytes just swept the floors. But recent findings have completely blown that open. Astrocytes are incredibly active participants in neural signaling. And in the cortex, THC activates CB1 receptors on these astrocytes.
But wait, this is where it flips, right?
Yes. Here's the massive paradigm shift. Unlike the CB1 receptors on neurons, which usually couple to those GIO proteins to act as a circuit breaker and inhibit activity, the CB1 receptors on astrocytes uniquely couple to a different protein, GQ proteins.
Okay, let's translate that again. While the neuron's receptors are acting like a brake pedal via GIO, what is this GQ coupling doing on the astrocyte? Is it acting like a gas pedal?
That is exactly right. When THC binds to these astrocytic receptors, the GQ coupling triggers intracellular calcium transients, massive sweeping waves of calcium inside the astrocyte itself, and this calcium surge prompts the astrocyte to actively release glutamate, the brain's primary excitatory neurotransmitter, right into the surrounding synapses.
So instead of shutting things down, the astrocyte is throwing fuel on the fire.
Literally. This burst of glutamate from the astrocytes further activates the frontal corticostriatal pathways. It adds this intense, stimulating layer to the rewarding effects of the drug. The city isn't just turning off traffic lights, it's rerouting massive amounts of power to specific, highly excitable districts.
And this overwhelming flood of dopamine, combined with this highly abnormal release of astrocytic glutamate, leads to one of the most bizarre and universally reported side effects of cannabis consumption.
The time distortion.
The illusion of time, exactly. I'm sure you, listening to this, have either noticed this yourself or heard a friend talk about it. Under the influence of cannabis, time seems to stretch out endlessly. A minute sitting on the couch can feel like an hour. The song you were listening to feels like it has been playing for 20 minutes. Have you ever wondered why that is? The slowing of time isn't just a subjective philosophical illusion, it is a literal mechanical disruption of the brain's timekeeping machinery.
Time distortion under THC is driven by a triad of neurobiological failures. 3 distinct systems breaking down simultaneously. First, as we discussed earlier, the basal ganglia loses its internal rhythm because its circuitry is heavily suppressed by THC. The brain's internal metronome is literally ticking slower.
Okay, so the clock is physically broken. What's the second failure?
The massive increase in dopamine. Dopamine governs salience, how important something feels. When dopamine floods the system, it makes each individual moment feel hypersalient and intense. It expands the subjective weight of the present moment. You are feeling so much in a single second that your brain assumes more time must have passed to contain all those feelings.
And the third failure brings us back to the hippocampus, which is failing to encode short-term memory.
Correct. Because your brain isn't recording the sequence of events properly, you lose those temporal anchors. You can't remember the beginning of your own thought, let alone the context the last 5 minutes.
So when the top-down part of your brain tries to calculate how much time has passed, it looks for memory anchors, finds none, feels the massive emotional weight at the present moment via dopamine, and relies on a broken basal ganglia metronome. The result? It overestimates time wildly. The internal clock is completely out of sync with reality, and that is just staggering to me. It really shows how fragile our perception of reality truly is.
It does. And the profound nature of these acute effects naturally raises a critical question for researchers and users alike. What happens when the brain is exposed to this hack chronically? The brain is a highly adaptive system. It will not simply tolerate a constant state of massive disinhibition, receptor flooding, and temporal distortion. It attempts to defend its homeostasis at all costs, leading to significant neuroadaptations.
Right. The city fights back against the hack. The most immediate defense mechanism the brain employs is called CB1 downregulation. To protect itself from this constant, overwhelming overstimulation from THC, the brain literally removes the CB1 receptors from the surface of its neurons.
It hides them.
Exactly. It puts the traffic lights inside the buildings where the THC can't reach them. The sources you provided highlight some incredible imaging studies showing exactly this.
Yes, these are high-resolution PET scan studies. PE stands for positron emission tomography. In these studies, researchers use a very specific radioactive tracer, or radioligand, called 11C-OMR. This tracer binds specifically to CB1 receptors, allowing us to physically see receptor density in a living human brain.
And what did these scans reveal about chronic daily cannabis users?
They show that chronic daily users have a 15% lower CB1 receptor availability compared to healthy non-users, particularly across the cortical regions. The receptors simply aren't there on the surface anymore.
If we connect this to the bigger picture, a 15% reduction across the cortex is a massive structural shift. And the adaptations don't stop with the endocannabinoid system, do they? The brain also takes aim at the reward system. Because the mesolimbic pathway is being artificially surged by THC, the brain reduces its baseline dopamine production and downgrades its dopamine receptor sensitivity. Over time, those massive dopamine surges start to blunt.
The system builds a tolerance. This creates what we call a hypodopaminergic state, a state of abnormally low dopamine function. And this biological reality maps perfectly onto the psychological symptoms we see in cannabis use disorder.
When the dopamine system is turned down, the user experiences anhedonia. It's often described as an amotivational state. Everyday natural rewards that used to bring joy, like eating a good meal, getting exercise, achieving a goal at work, or just hanging out and laughing with friends, suddenly feel completely flat and uninteresting.
The brain has adjusted its baseline so drastically that only the drug can provide a sense of normal motivation or pleasure.
It is a chemical numbing of the human experience. And beyond just receptor changes, there's evidence of physical, structural damage with chronic heavy use. The animal models detailed in our sources demonstrate actual physical shrinkage of the dopamine-producing neurons down in the VTA.
Yes. Furthermore, there is a documented loss of dendritic spines on the medium spiny neurons within the nucleus accumbens.
Dendritic spines, those are the tiny branch-like protrusions where neurons actually receive incoming signals from other cells, right?
Exactly. They are the physical connection points. Losing them means the network is literally losing its connectivity. The wires are fraying.
And this loss of connectivity creates a massive cognitive conflict within the individual. Your brain normally operates in a careful balance between top-down executive control and bottom-up impulses. The prefrontal cortex, or PFC, is the top-down manager, the mayor of the city. It handles reflection, impulse control, emotional regulation, and long-term planning.
And the basal ganglia and reward centers are the bottom-up drivers of immediate desire and habit. Chronic THC use visibly reduces the volume of the orbitofrontal cortex, a key part of the PFC, and creates what is termed a hypoglutamatergic state.
Glutamate, as we discussed, is the primary excitatory neurotransmitter. It is essential for the rapid, highly complex firing required for top-down executive control. When the PFC is deprived of glutamate signaling, the top-down manager, the mayor, is effectively asleep at the wheel. The individual loses the neurological ability to step back and reflect on their actions.
This drives highly impulsive behavior. It creates a powerful bottom-up urgency to use the drug to escape any negative emotions. Why? Because the very reflective circuitry required to regulate those emotions without the drug has been structurally weakened. It becomes this vicious cycle where the very tool the brain uses to say no is the tool being dismantled by the drug itself.
Is the defining trap of addiction. And this leads us to the critical threshold. What actually happens within the brain when the drug is finally taken away?
It's a profound transition.
There's a moment many people describe when they stop using cannabis. It's not dramatic, it's not loud, it's actually very quiet.
Yeah. And that quiet can feel strange, because for a long time the brain has been receiving the signal from THC.
When someone stops, the brain suddenly has to operate without that signal. The dopamine spikes are smaller, motivation can feel lower, even simple things may feel flat.
And that moment is where many people think something is wrong with them.
But often it's simply the brain recalibrating.
The brain is incredibly adaptive. When THC repeatedly activates the endocannabinoid system, the brain adjusts.
And when the signal disappears, the system slowly learns to balance again.
It doesn't happen instantly, but the brain begins rebuilding its natural dopamine rhythm.
And that's something fascinating about the brain. It can adapt to substances, but it can also adapt to freedom from them.
Which means the story doesn't end when someone quits cannabis.
In many ways, that's where the brain's real recovery story begins.
The biological reality behind that quiet we just described is intensely complex, and it isn't always peaceful. What is happening during this initial phase of abstinence is best explained by the opponent process theory of addiction.
Right. When the brain is chronically exposed to a drug, it pushes back. It mounts biological countermeasures to oppose the drug's effects and try to maintain its homeostasis. When the individual suddenly stops using, the drug is removed. But the brain's massive pushback is still happening. Only now it is completely unopposed by the drug.
This unopposed pushback is what triggers cannabis withdrawal syndrome, or CWS. For decades, there was a myth that cannabis didn't cause physical withdrawal. We now know that is categorically false. It isn't just a psychological desire for the drug. It is a profound physiological crisis of adaptation. The brain's central stress response system, known as the HPA axis, becomes highly dysfunctional.
Wait, before we talk about the panic of withdrawal, let's define the HPA axis for a second so everyone is on the same page. That stands for the hypothalamic pituitary adrenal axis, right? Basically, it's the body's central alarm system that controls how we respond to stress.
Yes, it is the feedback loop that dictates your stress levels. And without the suppressive, dampening effects of THC that the brain had grown accustomed to, this HPA axis goes into overdrive. The brain releases a massive surge of corticotropin-releasing factor, or CRF.
And CRF is the brain's primary stress chemical. This massive surge is responsible for the intense anxiety, the profound irritability, the vivid nightmares, and the severe sleep disturbances that define the acute withdrawal phase.
The brain is essentially in a state of chemical panic. It is trying to operate a system that has had its primary inhibitory receptors, the CB1 receptors, downregulated and hidden away, while simultaneously dealing with blunted dopamine levels and a flood of stress hormones. It feels awful because biologically the system is temporarily broken.
But the incredible news, and the real hope within the research you gathered, is the timeline of cognitive restoration. The brain possesses a staggering capacity for neuroplasticity. The recalibration process is arduous, certainly, but it is highly effective. Let's look at the early phase of recovery, spanning days 1 through 28. The speed at which the brain begins to heal its endocannabinoid system is nothing short of remarkable.
It really is. Remember those PT scans using the 11C OMAR tracer that showed the 15% downregulation in chronic users? Researchers used those same scans to track recovery. Within just 48 hours of monitored abstinence, the scans show a significant, visually measurable upregulation of CB1 receptors.
2 days. That's all it takes for the brain to realize the flood is over. The brain immediately senses the absence of the exogenous chemical and begins pushing those receptors back up to the surface of the neurons to capture whatever natural endocannabinoids it can produce. It's putting the traffic lights back on the streets.
Exactly. And within the first week, many individuals report that the mental fog begins to lift. Clinical research shows that verbal learning and memory capacity demonstrate dramatic improvement during these first 7 days.
And this correlates perfectly with the residual THC finally washing out of the fat-rich tissues of the hippocampus, right? Once the THC is gone, those sharp wave ripples and theta oscillations, the brain's internal recorders, can begin syncing up again. The temporal anchors return.
By day 28, the receptor density in the vast majority of cortical regions has returned to levels that are statistically comparable to healthy, non-using individuals. At this 4-week mark, the acute physical withdrawal symptoms driven by that terrible CRF stress surge have largely dissipated. The alarm system has turned off. However, it is vital to note that the hippocampus may take a bit longer to fully upregulate its receptors, which explains why some subtle memory issues can linger past the first month.
So what does this all mean for the person going through it? It means that the physical hardware, the CB1 receptors themselves, heal incredibly fast. But the software, the delicate rewiring of the deeper emotional and decision-making circuits, is a much longer journey. This brings us to the intermediate and long-term recovery phase, from month 2 up to a year.
While the receptors return to the surface quickly, the underlying synaptic plasticity, the actual strength and health of the connections between neurons, takes significantly longer to repair. The deficits in complex executive functions, sustained attention, and inhibitory control, those top-down prefrontal cortex functions we discussed, can endure for 3 to 4 weeks and often take up to 2 or 3 months to fully normalize.
The mayor needs time to get back into the office and start managing the city again. The brain literally has to rebuild the glutamatergic pathways that allow for reflective thought and impulse control.
It is a gradual rebuilding process, and then there is the dopamine system. The recovery of the dopamine baseline is what finally cures that heavy, quiet flatness we talked about in the opponent process phase. By month 2 or 3, the dopamine system begins to genuinely normalize. The nucleus accumbens slowly regains its sensitivity.
It begins responding effectively to natural rewards again. The joy of going for a run, the pleasure of a deep conversation, the emotional engagement with a piece of music. These natural reinforcers start to feel genuinely good again because the dopamine circuitry has finally shed the blunting effects of chronic use. The brain doesn't need the massive artificial flood to feel joy anymore. It can appreciate the gentle natural tides again.
It is a profound testament to the resilience of the human central nervous system. The brain can completely re-architect its reward and signaling pathways to accommodate a massive chemical intrusion, and then, given enough time and abstinence, it can systematically dismantle those exact same adaptations to restore its original healthy homeostatic balance.
Let's bring this all together. We have journeyed from the tiny electrical spark of a single action potential, relying on the tireless, energy-hungry work of the sodium-potassium pump, to the elegant circuit breaker of retrograde signaling via the endocannabinoid system. We've seen how a single molecule, a molecular master key called THC, can hijack this system. We watched it flood the locks, inhibit the GABA brakes, and surge dopamine, all while simultaneously distorting our very perception of time by disrupting the basal ganglia and the hippocampus.
We tracked the heavy, sometimes hidden toll of chronic exposure, the downregulation of receptors, the blunting of the reward system, and the physical pruning of dendritic spines that leads to intense cognitive conflict and impulsivity. But we also mapped the beautiful, complex machinery of recovery, watching receptors reemerge in a mere 48 hours and dopamine systems reawaken over a span of months.
It is a profound story of chemical manipulation and deep biological resilience. And it leaves us with something quite incredible to think about regarding how our minds actually construct reality.
Indeed. Our brains possess this ancient, highly intricate endocannabinoid system entirely dedicated to keeping our physical and emotional reality in a state of perfect balance. If a single plant molecule can so profoundly hijack this system to completely alter our perception of time, our encoding of memory, and our capacity for joy, what other hidden master regulators exist within us, silently constructing the reality you are experiencing right now, just waiting to be understood.
Keep questioning the world inside your own head. The architecture of your reality is more complex, more adaptable, and more fragile than you could ever imagine. Thank you for joining us on this deep dive.